When you are rubbing one body with another body, electrons are transferred from one object to what? Another object. Okay, this process, this type of electricity is been given a name.
Halogen connected carbon sp3, then it's connected to benzene. See all of you, halogen connected. What is this carbon hybridization? Full single bond sp3, see then a benzene ring.
Denominator poles shouldn't be included in the denominator poles. That means the denominator, these -1 and -5 should never be included in the solution.
Homocircle caudal fin. It's giving two equal lobes. But in case of cartilage fishes, look at here, one big lobe is there, one small lobe is there. You cannot cut into two equal half this fin.
Hi all, today I am going to discuss some important topics from sexual reproduction in flowering plants. So let's have a discussion about all the topics in this chapter with the questions also, especially the MCQ areas. Let me start the chapter with the first question. The following figure represents a flower, dash flower, and identify A, B and C. So here the given diagram from NCERT textbook, you may be knowing the structure of a flower. So let's have a discussion about the structure of a flower and what are the parts that are given here. You already know that the flower is attached to the stem or branch with the help of a stalk-like structure. That stalk-like structure is called as pedicel. And the tip of the pedicel will be becoming swollen and it will be forming the base portion of the flower. The base portion of the flower is called as thalamus or receptacle. And on this thalamus or receptacle, you can see they will be having nodes and internodes. Internodes are reduced or absent. Outermost node, you can see the leaves are actually modified into a layer. So outermost node that is coming in the flower, that will be calyx. Single units are actually called as sepals. Main function of this sepal is protection of the flower in bud stage. Then next inner to that, next wall you can see they will be having the beautiful colorful part of the flower that is actually called as corolla. Single units are called as petals. Main function is attraction of pollinators. Anyway, they are not involved in the gamete formation and all. So they are called as non-essential walls of a flower. Inner to that, there will be the male reproductive part of a flower. That is enriching. Single units are called enriching. The single units are called as stamen. And it consists of a thread-like structure, that is the filament. And at the tip you can see there is anther. Then at the center there is female reproductive part of a flower, that is gynoecium. And in the gynoecium you can see the single units are called pistil or carpel. If it is one carpel, we call it monocarpellary, many carpel, multicarpellary. And if the carpels are fused, it is called syncarpous. And if the carpels are free, that is called apocarpous. So I will come to that examples also. The pistil or carpel consists of a swollen bottom. That swollen bottom is called ovary. Inside the ovary, there can be ovule. Elongated part is called as style. And at the tip, there is a stigma. So this is the structure of a typical flower. Now let's go through this. Here you can see in the given diagram, it is having the A represent. This is the ovary, style and stigma. So A represents stigma, then B represent the sepal. The B will be representing the sepal and C will be representing the ovary. So here there is ovary also. So it's a bisexual flower. You can see there is stamens also. So stamen and pistil is present. So the answer is B. It's a bisexual flower where A represents stigma, B represents sepal and C represents ovary. So from this area, when you are learning, make this flow chart also. It will be very helpful. So understand that a flower is attaching to the branch or stem with the help of a pedicel. The stalk portion is the pedicel. The swollen end of that pedicel is thalamus or receptacle. On the receptacle, the leaf is modified. Flower is a modified shoot. So you can see the leaf is modified on each node. And in that outermost node, you can see the leaf is modified into calyx. Single unit is sepal. Main function is protection in bud stage. Inner to that there is corolla. Single unit is petal. Main function is attraction of insect. Inner to that there is male reproductive part, that is enriching. Single units are called stamen and it will be having a thread-like structure called as filament. At the tip there is anther. Then there is the female reproductive organ, that is gynoecium. Single units are called as pistil or carpel, where there is a swollen bottom, that is ovary, elongated part is the style and at the tip there is stigma. Hope you got this concept and the question. Shall I go to the next question? Please comment. Now coming to the assertion reason type question. So here, some plants shed pollen at 3-celled condition. Assertion, reason, these plants carry 3 male gametes since the beginning of reproduction process. So let's have a discussion about microsporogenesis. Then we will come back to the question. So here in the microsporogenesis, you can see the structure of the stamen. If you are taking the structure of stamen, if you are taking, there will be a filament and the filament will be having a connective, and on either side of this connective, so here a connective will be coming. On either side of the connective you can see it will be having a anther. If I take the cross section of anther, it looks like this. So this is the cross section of the anther. You know what is the outermost layer that will be coming in the anther. So at the center, the connective will be present and the outermost layer that is coming in the anther, that will be epidermis. And inner to this epidermis, they will be having the next layer that is present. So here, inner to this epidermis, there will be the next layer that is present. The next layer that is present inner to the epidermis will be endothecium. So there will be epidermis. Inner to the epidermis, the next layer that will be coming in an anther is endothecium. Then inner to that endothecium, there will be another layer that is coming. That layer that is coming inner to the endothecium, that is middle layer. So I am not drawing it completely. Then inner to the middle layer only there is tapetum. So like this you can see tapetum will be coming. So this is the cross section of an anther. So if you take the cross section of an anther, it will be looking like this. It will be having epidermis, endothecium, middle layer, tapetum. Inner to the tapetum only sporogenous tissue is located. That sporogenous tissue, they can actually divide and they will be giving rise to microspore mother cell. So inner to the tapetum present is sporogenous tissue. Now this sporogenous tissue will be turning into, it is diploid, it will be turning into microspore mother cell. This microspore mother cell which is diploid, they undergo meiosis and it will result in the formation of 4 haploid spores. Like this, 4 haploid microspores would be formed. And later this microspores will dissociate and they will be forming like this 4 microspores, that is haploid. And each microspore is now in a single celled stage. They will further divide and they will turn into a bicelled stage, where you can see there is a big cell and its nucleus, that is called as vegetative cell, and there is a small cell and its nucleus, that is called as generative cell. So understand, generative cell will be deriving nutrients from the vegetative cell. This is bicelled stage. In 60% angiosperm, they release this pollen grain. So this is the pollen grain which represent the male gametophyte. So 60% angiosperm, the pollens are released in bicelled stage. And in 40%, that only is coming in the assertion, some plants shed pollens at 3-celled condition. That is the remaining 40% angiosperm, you can see the pollens are released from the anther during dehiscence in the 3-celled stage. 3-celled stage means the generative cell will further divide and it will be forming 2 male gametes. So 3-celled stage, you can see it will be having a vegetative cell and its nucleus. In the cytoplasm of vegetative cell, 2 male gametes will be floating. So this is the 3-celled stage. So 40% angiosperm, you can see the pollens are shedded in 3-celled stage. So assertion is correct. Now about the reason. These plants carry 3 male gametes. You can see it's not 3 male gametes, it is only 2 male gametes. So one question. I have said this much. So one question, you should comment the answer. How many meiosis is required to form 400 male gametes? The number of meiosis required to form 400 male gametes. Can you show the comment box? Vishnu, comment box. The question, number of meiosis that is actually required to form 400 male gametes. Here you should understand, one pollen has 2 male gametes. So 400 male gamete formation, it will be requiring 200 pollens. So don't take it as 100, majority will be answering it as 100. No. 100 meiosis will be forming 400 pollens. We don't need 400 pollens. Each pollen has 2 male gametes. So the answer is not 100, the answer is 50. 50 meiosis will give rise to 200 pollen. Each pollen will produce 2 male gametes. So forming 400 male gametes require only 200 pollen and the number of meiosis required is 50. So this is an application question. Generally students make this mistake. So I think one student already commented, Dream has already commented 100 meiosis. That is actually not correct. It is 50. So you should understand the concept. It's an application question. So I asked not the number of pollen. I asked about the number of meiosis. So let's move to the next topic and the questions. So here the reason is actually wrong and assertion is only correct. So assertion is correct and the reason is wrong. Now next question. Anthers in angiosperms are bilobed and diathecous. So it is bilobed. 2 lobes are actually coming in the angiosperms. And each lobe will be having 2 pollen sac also. It will be having 2 sacs. So it is diathecous. Anthers in angiosperms are bilobed and it is diathecous. That is correct. They possess one theca in each lobe. That is wrong. It has two theca in each lobe. So here assertion is correct and the reason is wrong. Next one. Next question. Okay. Next one. Choose the correct match. Germ pore. You know what is germ pore? If I take the structure of a matured pollen grain, the outermost layer is actually called exine. And you know this exine is made up of sporopollenin. And at particular location you can see this sporopollenin is absent. That only we call it as germ pore. So germ pore is the region in the exine where the sporopollenin is absent. To that only you can see there is the pollen tube growth. So germ pore, it is exine. Then tapetum. Tapetum is the innermost layer that is coming and it has more than one nucleus. So when we were discussing about this structure, you should understand this innermost layer surrounding the sporogenous tissue is tapetum. This structure is tapetum. So you should understand the cross section of anther when you are taking outermost layer is epidermis. Inner to that there is endothelium. Next is middle layer. Then the next is tapetum. Tapetum is a nutritive tissue. They are multinucleated. They only provide nutrition to the sporogenous tissue and the developing pollens. They provide nutrients, they also secrete enzyme, and they also produce hormones that are essential for this microsporogenesis that is occurring. So tapetum in this, if I am taking, it will be cells with more than one nucleus. It will be cells with more than one nucleus. Then vegetative cell. Vegetative cell, irregular shape with large nucleus. So the structure of pollen if you are taking, inside is vegetative cell with a large irregular nucleus. Okay, so there is a vegetative cell with a large irregular nucleus, vegetative nucleus in its cytoplasm only. So here it is irregular shape with large nucleus. Then in time chiefly composed of what? Cellulose and pectin, CABD. CABD. So here understand in its cytoplasm only the generative cell will be floating. So this is the generative cell. The generative cell only later undergo equal mitosis to form two male gametes. So this is the structure of a typical pollen, that also you should understand. So a pollen structure if you are taking outermost layer is exine, and exine is made up of the toughest material that is sporopollenin, and inner to that there is intine. That intine layer is made up of cellulose and pectin, and there is a big vegetative cell and its nucleus. In the cytoplasm there is a generative cell also, and this generative cell only later undergo division to form two male gametes. So 40% only will be shedded in three-celled stage. Remaining 60% angiosperm you can see they are released in the two-celled or bi-celled stage. Hope you got the question. Yes. Yes, you are correct. Option 2 is the answer. Then next one, size of a pollen grain. So if I take the size of a pollen grain that is given in NCERT, it's coming around 25 to 50 micrometer. That is the size of a pollen grain. Then select the correct order of the wall layers from outside to inside in a microsporangia. The anther structure we were discussing, in that microsporangia, and there is microsporangia, it will be having 4 layers coming. Outermost layer is epidermis, inner to that, so epidermis is a single layer, protective layer. Inner to that is endothelium, that is also a single protective layer. Inner to the endothelium is middle layer. That diagram only I was drawing in the beginning. So same diagram only you have to learn. Outermost layer epidermis, that is a single protective layer. Endothelium is also one layer, and they only help in protection and dehiscence, breaking open of anther to release the pollen grains. Then inner to that is the middle layer. Middle layer you can see 2 to 3 layer of cells will be coming. Anyway, From this middle layer, tapetum keep on absorbing what? The nutrients. And they keep secreting it into the pollen sac. That is what is happening. So when this anther is maturing, you can see the tapetum and middle layer will degenerate. So in one lobe, instead of 2 sac, you can see only 1 sac, because the middle layer and tapetum will be degenerating towards the end. So hope you understood the 4 layer. Outermost is epidermis single layer, inner to that endothelium single layer, middle layer is multi-layer, 2 to 3 layer of cells will be coming, tapetum single layer, but all cells are multinucleated. So what is the answer to this? Outermost epidermis B, inner to that endothelium D, then middle layer, then tapetum. So it is BDAC. So the answer is okay. Reem Alam, the answer is correct. Tiwari Vedant also, the answer is correct, already answered. Now let's see the next question. Pollens have 2 prominent walls which are dash and dash. This already we discussed. Can you please comment? So I'm waiting for the comment. We already discussed what is the outermost layer and what is present inner to that. So outermost layer is, yes Reem, the outermost layer is exine and inner to that is the intine. So all of them got the answer, very good. Now next one. External factors affecting pollen viability. The time period up to which the pollens retain the capacity of germination, that is actually called as pollen viability. So mainly it is affected by temperature and humidity. Understand, when there is an increase in temperature and so much water is actually lost, the pollen will not be able to germinate. So if they are getting an optimum temperature with a good water content only, they will start germinating. So temperature and humidity is a factor that will keep them viable. Next is, consider the following and choose the haploid cell. Pollen mother cell is diploid, that is formed from the sporogenous tissue. They only undergo meiosis. Cell of pollen grain, vegetative cell, generative cell or the male gametes, they are haploid. Cells of connective, in the anther, epidermis, endothelium, middle layer, connective, all these if you are taking, they all are diploid. Then sporogenous tissue, that is also diploid. So ploidy based question also you can expect the answer is B only. Next one. The proximal end of the filament is attached to, many of them make mistake in this question. So here in a flower you can see there is sepal, then there is petal, and to that only the stamen is attaching. So the stamen can attach either to the thalamus or they may be attached to the petals. Proximal end means the starting point. So stamen's proximal end will be attaching either to the thalamus or to the petals. I think you might have learned morphology chapter last year, so in that you might be learning about epipetalis. In case of Solanaceae like family, the stamen's proximal end will be attaching to the petals. That term is epipetalis. Yes, have you all got it? Now next question. Which of the following has proved helpful in preserving pollen as fossil? For fossil forecast and all, pollens has been used in that. The pollens are actually getting preserved for a long time because of the covering that the pollens are having in the exine. And you know that toughest material. Which is that toughest material? Can you comment? Yes, Tiwari, the answer is sporopollenin. So exine, you should understand the pollen grains are having an outermost covering, that is exine. It is made up of sporopollenin. Then next one, consider the following statement and choose the wrong. Innermost layer, tapetum, nourish the developing microspore. That is the main function of tapetum. Epidermis endothelium protection. From middle layers, nutrients are absorbed by tapetum and they only secrete them into the sporogenous sac or the pollen sac. So main function of tapetum is nourishment. Not only that, they also release enzyme that is helpful in microsporogenesis. So they secrete one enzyme named callase. That callase enzyme only helps in the dissociation of microspore tetrad or pollen tetrad dissociation, that is with the help of callase enzyme, that is also secreted by tapetum. Then outermost layer of the pollen, that is actually made up of sporopollenin, that is also mainly secreted by the tapetum. So first statement is correct. Then second statement, flower is the main reproductive organ in angiosperm. They are kept under phanerogamy. Phanerogamy is flowering plants where the sex organs are visible. So that statement is also correct. Angiosperm, the main reproductive part is flower, which is a modified shoot. That only we have discussed in the beginning. Flower is a modified shoot where the shoot is modifying into pedicel, thalamus, leaf modifying into sepal, petal, etc. The next one, when the anther is young, a group of compactly packed arranged homogeneous cells called sporogenous tissues are present and they turn into megasporangia. It's not megasporangia. Actually the statement, if I take this part, is actually wrong. It's not megasporangia, it is microsporangia. So the third statement is wrong. Now let's see why the fourth statement. Stored pollens in liquid nitrogen can be used in crop breeding program. So pollens can be preserved for a long time in liquid nitrogen at minus 196 degree Celsius. So there is the pollen banks and all where you can see the pollens can be preserved for a long time, because viability of pollens, if you are taking, it is different for different plants. Some pollens are viable for only 30 minutes. Some pollens you can see they can be viable for few months also. So like that viability time or the germination time or the time period up to which pollens retain the capacity of germination, that is different for different pollens. So pollens are stored in liquid nitrogen that is used for crop breeding program. That statement is also correct. So only third statement is wrong. Next question, sporopollenin. We already discussed this. Sporopollenin is the outermost layer of the pollen grain that is found in exine. It is the toughest material. Is the major component of intine is wrong. Can be degraded by enzymes. No, it cannot be degraded by enzyme. They are highly sensitive to increased temperature and environment. They can withstand high temperature and all. So they can withstand acids and alkali. So understand, this is the toughest material and it will be protecting the pollen. Next one, choose the correct match. Monocarpellary. So when you are taking a flower and if it is having a single carpel like this, you know carpel consists of a swollen bottom, that is ovary, and inside the ovary they will be having a ovule, and elongated part is called as style, and at the tip they are having a stigma. So like this, if only one carpel is present, it is called monocarpellary. So monocarpellary is single pistil or carpel. Then multicarpellary, many pistils or carpels are present, it is called as multicarpellary. So some flowers you can see many pistils or carpels are present. In that itself, if they are fusing, then it is called syncarpous. So for example, if you are taking the flowers of Solanaceae or else Liliaceae, Brassicaceae, all these you can see it's multicarpellary. More than one carpel is present and these carpels are fusing together. So if you look at them, you may feel it is a single carpel, but it's not. It's actually a fusion of many carpels. But in lotus, rose, michelia, etc., if you are observing, you can see these carpels are free. In a flower, many carpels are present and all carpels are free from each other. That condition is called as apocarpus. Free pistils are coming, that is called apocarpus. CABD. So this is the answer. Hope you got it. Yes, it's option 2. Now next one, analyze the figure and find the correct labeling. Can you do it? So it's the structure of embryo sac that represent the female gametophyte. So the development of embryo sac, let's have a discussion. From one of the cell of nucellus, there will be formation of megaspore, mother cell. Hope you remember the structure of a typical ovule. So inside the ovary, there is an ovule that is coming. So in the ovule, you can see there is a funicle. There is a stalk-like portion that is coming in the ovary, that is called as funicle. The point of attachment is called as hilum. And from there, you can see there is the base portion of the ovule that is called as chalaza. Then next you can see there is an elongation coming from chalaza, that is the integument. So there is an outer integument and there is an inner integument. You should understand this outer and inner integuments only become the seed coat. They are the future seed coats. Then this region, that is the micropyle. So there is micropyle. Now here there is some cells coming, that is nucellus, and in that one cell only is turning at the center into the megaspore mother cell. So this megaspore mother cell which is diploid, they will be undergoing meiosis. So let me write it down. This is the megaspore mother cell. They will undergo meiosis, and this meiosis will be resulting in the formation of 4 megaspore. In that 4 megaspore, 3 will degenerate. 3 of the megaspores will degenerate. 1 will become functional. So 1 will become functional megaspore. And remember, this functional megaspore is located towards the micropylar end. Then they are actually facing towards chalaza at the micropylar end. That is what you should understand. Here only 4 will be formed. 3 towards the micropyle will degenerate. 1 at the micropyle facing towards chalaza will become functional megaspore. In that functional megaspore, you can see there is nuclear division. So first round of nuclear division, they will form 2 nucleus. Then this nucleus will be moving towards each pole. So this is the first round of nuclear division. After moving towards each pole, they will undergo second round of nuclear division in order to form 4 nucleus. Then they will undergo third round of nuclear division in order to form 4 nucleus at each poles. Then finally, after 3 round of nuclear division, they will be turning into the functional embryo sac. So after this, you can see there will be 3 nucleus at the chalaza, 3 nucleus at the micropyle, and 2 nucleus at the center. This is followed by cytoplasmic division, cytokinesis. So 3 nucleus that is coming at the chalaza will be turning into antipodals. The 3 nucleus coming at here will be turning into antipodals. Then 2 that is coming at the center from each pole, they will be turning into polar nucleus. So A is antipodal, B is polar nucleus, C will be the central cell. C will be acting as the central cell. D is the egg and E represents synergid, F represents filiform apparatus. So the answer will be, can you comment what is the answer? Tiwari has already commented, the answer is 2. So exact NCERT diagram only, hope you understood the megasporogenesis. So understand, inside the ovary, there is ovule. Ovule structure also try to understand. There is a funicle, hilum, chalaza, integument. Inner to that there is nucellus, and in that one cell is turning into megaspore mother cell. Megaspore mother cell undergo meiosis. They will form 4 megaspore. All these are happening here, whatever I have drawn here. And in that 3 of them will degenerate. One will turn into functional megaspore. Functional megaspore will undergo 3 rounds of nuclear division in order to form 8 nucleus. After that, cytokinesis will be occurring and they will turn into the functional embryo sac. So like this, development of embryo sac, the 7-celled 8 nucleated embryo sac, 7 cell, 3 antipodal, and there is 3 egg apparatus, 1 egg, and it will be having 2 synergids. Then at the center, the biggest cell is central cell with 2 nucleus, that is polar nucleus. Remember, all these are haploid. So there is 7 cell and 8 nucleus embryo sac. This is called polygonum type embryo sac. It is seen in majority angiosperm. And like this development of a functional embryo sac from a single megaspore is called the monosporic development. Sure question, monosporic development. Now, find the correct sequence of parts in an ovule from inner to outer region. So ovule structure just now we have drawn. So in that you have to tell which is coming from inside to outside. So if I take the structure of a typical ovule, there is a stalk-like portion in the ovule. That stalk-like portion in the ovule is called as funicle. Point of attachment, that is called hilum. Then the base portion of the ovule that is coming here, that base portion of the ovule is called chalaza. And there is an extension that is coming from the chalaza that form the seed coat in future, that is, what is that structure? Integument. There is an inner integument and there is also an outer integument. So there is 2 integuments are coming. There is a region where the integuments are not fusing. That region is called as micropyle. So understand, there is inner integument and there is outer integument. You know that after fertilization, ovule is becoming the seed. Remember, the integuments become seed coat in future. In that, inner integument will be turning into tegmen. That is the inner layer of seed coat. Outer integument will be turning into testa when an ovule is turning into the seed. Now, inner to this only, there is nucellus, the nutritive layer that is coming. Nucellus can be observed, that is present inner to this. So there is nucellus. Now inner to that nucellus only, a functional megaspore will be turning into an embryo sac. So near to the chalaza you can see there is 3 antipodals, and here there is a polar nucleus and the central cell. Here there will be synergids with the filiform apparatus and there will be also an egg. Hope you got it. So this is the structure of a typical ovule with the embryo sac. You should understand it is 180-degree inverted downward. That is called anatropous ovule. You can expect question from these areas also. So which type of ovule is seen in? Majority angiosperm. You can see majority of angiosperm like this, an inverted ovule can be observed. That is called anatropous. Upright ovule is there, that is called orthotropous. 90-degree ovule is there, that is called hemianatropous. But this one that is inverted downward like this, that is called as anatropous ovule. Majority angiosperm will be having anatropous ovule. So here inside to outside, egg, embryo sac, nucellus, integument. So which option, can you please comment, which is present from inside to outside? Okay, already it is commented. It is egg, embryo sac, nucellus and integument. Egg, embryo sac, nucellus, integument. That is from inside to outside. Now next question. consider the two statements and choose the right option. Self-incompatibility is a genetic mechanism which prevents autogamy as well as geitonogamy. So what is autogamy and geitonogamy? Let's have a discussion. Already you know. In a plant, within a flower, if there is transfer of pollen grains from the anther to the stigma, so from the anther to the stigma of a flower, if the pollen grains are falling on flower itself, so from the anther to the stigma, if the pollen grains are falling, that is called autogamy. Genetically and ecologically, autogamy is self-pollination. Then, if there is transfer of pollen grains from anther to stigma of different flowers of different plants, different flowers of the same plant, if like this it is falling, that means from the anther to the stigma of different flowers in the same plant it is happening, that is called geitonogamy. That is called as geitonogamy. So understand, if there is self-incompatibility, that is a genetic mechanism. So what is self-incompatibility? A plant will never accept its own pollens. If its own pollens are falling on the stigma of a flower, then they won't accept it. Pollens will not germinate. So self-incompatibility can be observed. That is a mechanism to prevent self-pollination and promote cross-pollination. Now, if both staminate flowers and pistillate flowers are present on same plant, the status is monoecious. Monoecious means bisexual. Understand, if a plant is having bisexual flower, then it is monoecious. And if a plant is having both unisexual flowers on the same plant, then also it is monoecious. So that is also correct. Both the statements are correct, both 1 and 2. You can see it is correct statement. Okay, Reem already answered. Hope you got it. So two terms you should understand: autogamy, geitonogamy. Transfer of pollen grains from anther to stigma of same flower in the same plant is called as autogamy. Transfer of pollen grains from anther to stigma of different flowers of same plant, that is called geitonogamy. That is ecologically cross-pollination, but I should tell it is genetically self-pollination. And if the pollen grains are transferred from anther to stigma of different flowers, anther to stigma of different flowers but of the different plants but of the same species, like that it is happening, this is actually called synogamy. Then two more statement, what is monoecious and dioecious. If a plant is having both unisexual flower in the same plant or bisexual flower, then it is called monoecious or bisexual. But if a plant is having only male flowers, staminate flowers in one plant, pistillate flowers in another plant, then it is called dioecious. Dioecious means sexes are separate. Now, enclosed within the integuments of a typical anatropous ovule is a diploid mass of cellular tissue. So what is that diploid mass of cellular tissue that is coming? The answer is nucellus. That is the nutritive tissue for the embryo sac. This is nucellus. This will be the diploid mass of cell that will provide all the nourishment to the developing embryo sac. Yes, very good. Mohammad Hisham has answered the question. Then next one. Pollen grains of rice plants lose their viability within minutes of their release. So you know how much time is taken for losing the viability in rice plants. Can you answer? What is the pollen viability in rice, wheat and all? How much time? It is 30 minutes. But in Rosaceae, Leguminosae, Solanaceae, etc., you can see they are viable for a few months. But in some plants, after release of these pollens, they are viable. They retain the capacity of germination only for a short time, like 30 minutes and all. Next one. Megaspore mother cell undergo mitosis to produce 4 megaspore. Already we have discussed megasporogenesis. Remember, megaspore mother cell will undergo meiosis in order to produce 4 megaspore. In that, 3 that is facing towards the micropyle will degenerate. 1 at the micropyle facing towards the chalaza will become functional megaspore, which undergo 3 round of mitosis or nuclear division followed by cytokinesis in order to form 7-celled 8-nucleated embryo sac. Then megaspore mother cell and megaspore both are haploid. Megaspore mother cell is diploid. After meiosis, they will form megaspore that is haploid. So both assertion and reason are incorrect. Next one. Embryo sac development is monosporic in polygonum type of embryo sac. I already said what is polygonum type. The 7-celled 8-nucleated embryo sac formed from a single functional megaspore that is called polygonum type of embryo sac. So here in this question, the assertion is correct. Then in this development, only one functional megaspore is present at micropylar end. That is also actually correct. So here you can see only one functional megaspore will be present. So both assertion and reason are correct, but reason is not the correct explanation of assertion. Actually, it is present at micropylar end facing towards the chalaza. Next one, which of the following is meiocyte? Meiocyte means the one which is undergoing meiosis is actually called as meiocyte. Can you tell the one which is undergoing meiosis? Microspore mother cell is undergoing meiosis and it is actually diploid. Functional megaspore is haploid. Haploid cell will not undergo meiosis. Generative cell is haploid. That is also not going to undergo meiosis. Central cell is also haploid. That will not undergo meiosis. Hope you got it. Now next one. Which is not a condition favoring cross-pollination? Can you tell the answer? The one which will not allow cross-pollination. I am asking about a point which will promote self-pollination. Yes, Muhammad Hisham, the answer is 4. Cleistogamy. You know about chasmogamous and cleistogamous flower. The flowers that are always remaining closed, that is called as cleistogamous. In these flowers, there is no chance of cross-pollination. Pollens from some other plants will not be falling here. They can only undergo cleistogamy, or in that they can undergo only self-pollination. So these plants are invariably autogamous. You can write that point. Cleistogamous flowers are invariably autogamous. They can undergo only self-pollination. So remaining whatever is written is outbreeding devices. That is the characters produced by the cross-pollinating plants in order to prevent self-pollination. So unisexuality, if a plant is having unisexual flowers, there is more chance of cross-pollination. And if they have bisexual flower, they will try to show what? Self-incompatibility. That means on pollens are falling on the stigma won't be accepting it. Then third, you can see anther and stigma are present at different levels. So that also will promote cross-pollination, or else the maturation of pistil and the maturation of anther will not be synchronized. That means the pollen release or pollen formation and release, as well as the embryo sac formation or maturation, it will be taking place at different time. So there is no chance of self-pollination. So the first 3 actually represent outbreeding devices, or the adaptations of cross-pollinating plants in order to prevent self-pollination. Whereas you can add one more point that will promote cross-pollination, chasmogamy, open flowers. Open flowers actually promote cross-pollination. So hope you got around 5 points regarding outbreeding devices: self-incompatibility, unisexuality, or diesy. Diesy is the term written in NCERT. Diesy means dioecious. then anther and stigma that is present at different level, then chasmogamy. These are the points coming under outbreeding devices. Now next one, consider the following statement. Have you got it? Can you please make a comment? Yes. Now, consider the two statements. The mechanism to discourage self-pollination and increase cross-pollination is called outbreeding devices. So, this is seen in naturally cross-pollinating plants. So they show this mechanism in order to avoid self-pollination, because if they undergo self-pollination, there is chance of losing the quality or vigor. That we call it as inbreeding depression. Remember that point also. Inbreeding depression is the loss of vigor or quality observed in naturally cross-pollinating plants when they are undergoing self-pollination. So if they undergo self-pollination, there is chance of losing the vigor or quality or becoming sterile. That is called inbreeding depression. So they adapt some mechanism to avoid this self-pollination. That is actually called as outbreeding devices. Second statement, dioecious plants prevent both autogamy and geitonogamy. Both statements are correct. Dioecious means unisexual. Sexes are separate. Male flowers on one plant, female flowers on another plant. There is no chance of autogamy and geitonogamy. Autogamy means what? The transfer of pollen grains from anther to stigma of same flower. Geitonogamy means transfer of pollen grains from anther to stigma of different flowers of the same plant. That is not at all possible in case of dioecious plant. So both the statements are actually correct. Next one, pollen tube generally enters into embryo sac through. So that part also let's have a discussion, especially double fertilization. A pollen after falling on the stigma through the germ pore, you should understand the vegetative cell will be coming out like this. And first exiting is the vegetative nucleus. That vegetative nucleus will be called as tube nucleus. Then following that only the two male gametes will be coming. So whenever a pollen is falling on the stigma of a plant, and if the stigma is going to accept this pollen, they will be releasing sugar and moisture. They will be uptaking the sugar when the water potential is decreasing inside the pollen grain. They will uptake also water or moisture. Then there is turgidity and there is growth of pollen or pollen tube through the germ pore. And through the germ pore, when this pollen tube is elongating, first exiting is the tube nucleus, that is vegetative nucleus, which will control the growth of pollen. And following that, two male gametes also will be exiting. Slowly they keep on growing and they will be reaching the ovary. And in the ovary, there is embryo sac. You know that it is getting covered by integuments. So like this, integuments are present, funicle is present. Through the micropyle only, there will be the entry of pollen tube. So pollen tube will be entering through the micropyle. And after entering here, you can see one of the synergids will be degenerating when the pollen tube is entering here. So pollen tube will be coming like this. Through the micropyle they are entering and they release the tube nucleus and they also release the two male gametes. By the time one of the synergid will degenerate. So when this synergids one is degenerating, then the pollen tube is entering. Anyway, the entry is guided by filiform apparatus in the synergid. So entry of pollen tube into the embryo sac is guided by filiform apparatus. Then one synergid will degenerate. And they release the tube nucleus and two male gametes. One of the male gamete fuses with the egg to form zygote. The process is called syngamy. Another male gamete fuses with the polar nucleus. But what you should learn also, let me tell. Inside the embryo sac, the central cell is having two polar nucleus. Two polar nucleus fuses to form secondary nucleus. To the secondary nucleus only the male gametes are fusing, and it will form a primary endosperm nucleus. So 2 haploid polar nucleus is there. This 2 haploid polar nucleus will fuse. So 2 polar nucleus will be fusing in order to form a secondary nucleus. Into the secondary nucleus only what is fusing? So secondary nucleus is diploid. Don't think polar nucleus is diploid. Polar nucleus is haploid. Secondary nucleus is diploid. To that secondary nucleus only the male gamete is fusing. When the male gamete is fusing, you can see it will result in the formation of, this will be resulting in the formation of a triploid primary endosperm nucleus. Now, the central cell will be called as primary endosperm cell. This process is called triple fusion. The process is called triple fusion. At the same time, the egg will be fusing with the another male gamete. So, another male gamete will be fusing with the egg. This will form zygote. The process is called syngamy. The process is called syngamy. So like this, 2 fertilization happens at the same time. This is called as double fertilization. The process is called as double fertilization. Hope you got this much. This is also an expected question area, double fertilization. So pollen tube's entry is through one of the synergates. One question let me ask. How many gametes are involved in double fertilization? How many gametes are involved in double fertilization and how many nucleus are involved in double fertilization? Gametes and nucleus, number only you type. How many gametes, how many nucleus? Let me check how many of you will get the right answer. So number of nucleus involved is 5. 2 polar nucleus, 1 egg nucleus, 2 male gametes. So 5 nucleus is involved. Number of gamete involved is 3. 2 male gamete and 1 egg. That is the answer. Now moving to the next question. Oldest seed, that is lupinus. Oldest seed is lupinus. Pseudocarp means false fruit, that is seen in apple and strawberry. Parthenocarpy. Parthenocarpy also is an important topic. Formation of fruits without fertilization. Some hormones can all induce the flowers to turn into fruit without fertilization. Especially the ovary will become fruit without fertilization. So these fruits will be seedless. Banana as well as some seedless orange, grapes, etc. You can see it is induced by some hormones. So here parthenocarpy, example banana. Then endosperm, tender coconut of water, that is an example of endosperm. BCAE. So this is the answer. Hope you got it. Clear? Next one, match the following. Apocarpus. Apocarpus means you can see many carpels are present and the carpels are free. That is seen in lotus, rose, michelia and all. So michelia, it is apocarpus. Then syncarpus. Syncarpus means you can see all the carpels are fusing together. That is actually called as syncarpus. So syncarpus example is hibiscus. Then outer integument. Outer integument is going to become testa. I said that integuments in the ovule will be turning into seed coat. Outer integument will become outer seed coat, that is testa, and inner integument will become inner seed coat, that is tegmen. Then ovary wall is going to turn into the pericarp. I think you might have learned this in morphology of flowering plants in grade 11. Pericarp is well differentiated in fleshy fruit. outer layer in mango, that is the epicarp that you peel out, and the middle fleshy layer that you are eating, that is the mesocarp, and the inner stony layer, that is the endocarp. Hope you got this much. Now next is about the structure of ovule. We already discussed the structure of ovule. So what is funicle? Funicle represent the stalk of the ovule. The stalk of the ovule is called as funicle. Then hilum. Region where body of ovule fuses with the funicle, that is actually called as hilum. Then integument. Integument, one or two protective layers of ovule is called as integument. If only one integument is present, it is called unitegmic. Example, gymnosperm. If two integuments are present, it is called bitegmic. Example, angiosperm. A typical ovule is bitegmic. Then chalaza, the basal part of the ovule from where integuments are arising, that is actually called chalaza. Then nucellus, mass of parenchymatous cells that will be providing nutrition to the developing embryo sac, that is actually called as the nucellus. Hope you got this much. So answer 5, 4, 3, 2, 1, that is 2. Then regarding the pollination, so here the pollination by wind, that is called as anemophily. So majority of the abiotic pollination, if you observe, that is by wind only, that is anemophily. In grasses and all, date palm and all, you can see it is anemophily is occurring. Whereas Vallisneria, Hydrilla, Zoostera and all, you can see it is pollination by water, that is called as hydrophily. So hydrophily is observed in freshwater plants like Hydrilla as well as Vallisneria. Then the marine plants like Zoostera, the longest pollen is observed in Zoostera, and character also you try to remember, they will be having a pollen that is getting covered by mucilaginous sheath. Then anemophily characters are also asked, that also you should learn. In plants that are pollinated by wind, they produce pollens that are lightweight, non-sticky in enormous amount, and they will be having a feathery stigma, tassels and all in corncob. The feathery stigma that will be waving in the wind, fluttering in the wind in order to capture the pollen grains. Then entomophily, that is pollination by insects and all, that is observed in rose, jasmine and all. Then begonia, you can see it is pollination by the birds. 1, 4, 2, 3. Hope you got this much. Yes, ream. Hope you got the answer. Next one. Which of the following option is correct? Transfer of pollen grains from anther to stigma of same flower. We already discussed, that is autogamy. Transfer of pollen grain from anther of one flower to stigma of another flower but in the same plant, that is gytinogamy. Then transfer of pollen grains from anther to stigma of genetically different plant of same species, that is xenogamy. So all the options are actually correct. Now endosperm is completely consumed by the developing embryo in, example is dicot. Generally you can see in dicot the endosperm is completely consumed. So what happens? Exceptions are there, but in dicot you can see generally during the development of embryo into the plumule, radicle, cotyledons and all, endosperms will get completely consumed. So such seeds, dicot seeds, are called as X-albuminous or non-endospermous. They are called X-albuminous or non-endospermous. Example of X-albuminous or non-endospermous seeds are pea and groundnut. Castor, you can see it's an exception. It is having endospermous seed. Then maize also, if you are taking, it is non-endospermous, like that, sorry, endospermous. So castor is an exception that you should learn. In albuminous seed, food is stored in. Albuminous means endospermous. Generally, monocot seeds are endospermous. Albuminous seeds, the food is stored in endosperm. Ex-albuminous or non-albuminous dicot seeds, generally, exceptions are the dicot seeds, it is stored in cotyledons. So that's all about today's question discussion and important areas from sexual reproduction in flowering plants. Hope it was helpful and you understood the session and enjoyed it. Okay then, bye.